Welcome everyone back to this cell signaling course. We'll start from where we left. If you recollect, in the last class we studied how quorum sensing was being regulated in terms of biofilm formation, also how certain bacteria use this two-component system for ensuring their life forms, or in some sense we can say how quorum sensing or any of these activities which bacteria engage in is tightly regulated by two-component system. The last example which we saw was for Caulobacter crescentus, which was undergoing asymmetric cell division and giving two types of cells. One had a swarmer fate and the other one was stalked. And we notice how two distinct types of a two-component system, one which involved the division kinase and the other one which involved the cell cycle regulation kinase. So div J and K as a simple canonical two-component system, and the other one involved CCKA and CTRA as a hybrid two-component system, where we notice that a cell which is going to become stalked cell has high div J activity, which was the kinase, which was phosphorylating the div K, which was the response regulator. So phosphorylated div K level is high in the stalked cells and low levels of phosphorylated div K were there in the swarmer cell, and this was ensuring that cells undergo cell division. And in addition, we had seen an example of how chemotaxis using the CHA kinase was used. We will take more example of this in our class today, where we will see how process like sporulation is regulated by two-component system, and we will take a very specific example of quorum sensing, where bacteria involved in the process emit light to signal each other about their presence, and this something is called bioluminescence. We will also notice that how some of the very complex processes which we see in higher eukaryote is also present in bacteria when they are signaling through two-component system. So let's see about the sporulation in Bacillus subtilis. Sporulation in microbes is generally a process which is induced depending on the cues which it is getting. Remember that any bacteria or any microbe for that matter never undergoes sporulation in a very rich environment, nutritionally rich, no stress and so on. So if nutrition levels or nutrient levels go down, you will notice that bacteria may decide to spore. If pheromone levels are there, so bacteria release certain compounds which can be considered as a pheromone, and they respond to that. If more bacteria is in the surrounding, they will release more of these components, and that will be sensed by bacteria. So if more bacteria around, which means that there is less of nutrition available because of the consumption, high consumption. So high pheromone levels, high chances of sporulation. Low nutrient levels, high chances of sporulation. Similarly, there are cell cycle cues. When cell is normally dividing and it does not face any stress, it will not undergo sporulation. But if there is a Q, or also if there is a damage to the DNA, then also the bacteria would avoid going into the sporulation, because they want to make spore which should survive the harshest condition. Similarly, if there is a replication stress, as I mentioned, then there will be a problem. The pheromone levels can also be connected with the population density. So all in all, you understand that bacteria is sensing the presence of nutrient, the pheromone, cell density, something which is telling whether it to divide normally or don't divide, undergo sporulation, or a stress which might be there because of the presence of some molecule, some entity which is affecting the replication process. What happens here is that many of the sensor kinases are involved in the signaling. If you recollect, we discussed that in signaling there could be multiple receptor boiling down to a single response regulator or effector molecule. So the sporulation process in Bacillus subtilis presents a case of that type, where multiple of the sensor kinases, which are briefly known, labeled as kin. So there are 4 or 5 of them, A, B, C, D and E. Multiple of them are there, they interregulate the process of the signaling, but we will discuss more about the kin A and B today. So these kinases are present at the cell surface and they will respond to the cues which will activate the pathway to sporulation. So as I said, they will be working towards activating the sporulation gene. The activity of this kin A or this kind of molecule is regulated by one of the things which I discussed earlier, that presence of an inhibitor. So what happens here is that the kin molecule, which is here, normally it is the kinase which auto activates itself in presence of right set of cues, which is towards the sporulation. So kin A undergoes phosphorylation and this is an autophosphorylation event, and the residue will be histidine here. But normally when there is sporulation is need not be present in the system, there is an inhibitor called KIP-I which keeps the kin A inactive, which basically means that kin A cannot undergo phosphorylation event in presence of KIP-I. In essence, kin A cannot undergo phosphorylation in presence of the inhibitor KIP-I. How, when this sporulation process have to be activated, what goes on there? Then you have another inhibitor. This is anti-inhibitor. And this is the inhibitor. If you recollect this, we discussed when we were discussing the regulation on cell signaling, where we say that inhibitor of inhibitor can be there to relieve any inhibition process. Same here happens, that when bacteria needs to activate the sporulation process, you will need kin A to be phosphorylated, and thus you will need KIP-I to be removed, and this KIP-I can be removed by its binding with KIP-A, and basically it will sequester the inhibitor and serving as a anti-inhibitor. Now let's see what happens basically inside the bacteria when this process must start. So there must be some signal which sense that there is a problem, or less nutrient available, or cell density is more. So in some sense, less nutrient available is like a starvation signal. So this starvation signal is received by the kinase, which is these two as shown here. Another kinases, kin C, kin D and kin E, they are also involved in interregulation. But let's understand, because the redundancy of their function, let's see how kin A and kin B are mediating this response. So you can understand that what exactly would starvation signal would be. Suppose it is the availability of glucose, a nutrient of that sort, or an amino acid. This receptor was not responding to the levels change in these nutrient, but when the nutrient level goes down because of excess population, these receptors are activated. These receptors are also kept quiet because if there is a replication issue or if there is a nitrogen availability issue. So together, these different conditions which are important for the growth of the bacteria, they ensure that the receptor is activated when the right set of cues are present. So what will happen? This is your sensor kinase, and as we know, the sensor kinase auto activates itself by phosphorylation on histidine, and then it transfers the phosphate group onto this response regulator, which is sporulation 0F. As a process, it will be activated further. Now this response regulator will continue. This is not the final response regulator. In one of the previous class, we said that there could be multiple of response regulator, and there is a master response regulator which is responsible for ensuring that signaling meets its end. So this SPO0F is the response regulator in sense that it has the receiver domain and that receiver domain will utilize this phosphate on its aspartate residue. Normally this response regulator which takes the signaling forward is inhibited by identifying the cell density. If cell density is less, it will be inhibited and there will be no sporulation. If cell density is high, this inhibitor will be removed and this SPO0F is active to take the signaling forward. So how it moves forward? You can sense that the histidine residue on this receptor is utilized. Now you have got the aspartate residue on the receiver domain where phosphorylation has happened. To move the signaling forward, you will need something which has the histidine moiety. So this is where you have histidine phosphotransferase. And this histidine phosphotransferase cannot be the master regulator. However, this SPO0A is the master regulator. So here again you have histidine which is phosphorylated, signaling moved forwards by the phosphorylay event, and the final master regulator SPO0A is phosphorylated on the aspartate residue and it is activated. What this activation leads to? In the activation of sporulation genes. You can easily understand when we say activation of sporulation gene, the property of SPO0A will be to have a transcription factor activity. So in this process, you have SPO0F, which is a minimal receiver domain containing response regulator, which passes the phosphate group to the histidine phosphotransferase and then finally to the master regulator. And this SPO0A now ensures that activation of sporulation genes happen. These signaling pathways are very tightly regulated by feedback mechanism. So some of the feedback mechanisms are here where you have the concentration of the bacteria in the medium, the availability of nutrient and so on. Also at multiple level there are regulation, like here the bacillus density and the inhibitor works on SPO0F rather than working on the receptor. So you can manage the signaling event at different steps. Similarly, the final master regulator which is there is regulated or inhibited by this SPO0E. So together you will see that multiple components come and they ensure that the signaling meets its goal, signaling which started at the cell surface by virtue of binding of a signal to its ligand, and in this cases the starvation signals were the one which started this entire process. Now let's look at another phenomena which happens in bacteria, and there is a class of bacteria which undergo bioluminescence. You can see a lot of videos which are available on YouTube freely showing that how in seawater there is light, luminescence happens, and that can be noticed particularly in the night. Particularly when somebody is moving or there is a disturbance in the water, this bioluminescence is even more. So what exactly is bioluminescence? And we will be taking examples of Vibrio harveyi. So bioluminescence is a consequence of quorum sensing. We have discussed quorum sensing in the context of biofilm formation. The idea was same here, is that you need a group of bacteria to come together and signal to each other and respond to each other. In previous case where biofilm was forming, these bacteria were coming together and they were holding hands together to form a film. What happens in quorum sensing and bioluminescence, that a threshold of bacteria when reached, they generally emit the signal, and each of these bacteria start responding to the presence of that signal or inducer, we will see as we move forward. It's a very complex kind of signaling which we'll understand as we go forward. So the molecules which is being released by these bacteria, they are all called autoinducer, and they sense the density of the cells in given volume. Now once there is a threshold which is reached, you will see bioluminescence happening. And it's a very great example of something which is initiated when the density is low. Basically the signaling is reversed in this case. We have been seeing that how signaling forward affects or meets the end. Here signaling basically will be reversed so that the bioluminescence can be seen. Now what happens over there in the case of bioluminescence, there are quorum related signal, if we can use it interchangeably, quorum / bioluminescence. So there are receptors which are named as lux basically to measure the light. So there are these signal molecules which are called autoinducer. AI stands for autoinducer. So there are different types of autoinducers which are produced, and these autoinducers, they work on their respective receptors. What you notice here again, that this is many to one kind of signaling, where three different receptors which are shown here, they are working through this response regulator and so on, which is in the lux family of genes. Lux U, Lux O, Lux R. So what you notice here is that the very first thing which is coming after the receptor is the histidine phosphotransferase, which talks about itself that it is a hybrid kind of receptor. So the receptor has the kinase activity, which will phosphorylate a histidine residue, and then there will be internal aspartate residue somewhere, and you will see the internal phosphotransfer event. Now from here, the next molecule which needs to take the signaling forward is the histidine phosphotransferase or Lux U. So when there is less number of bacteria and the autoinducers are less in concentration, these receptors engage in sensing the levels of bacteria and activate the pathway where Lux U is now receiving the phosphate residue, which means that signaling is moving forward. This Lux U now activates the Lux O, which is our response regulator. This response regulator in general is an inhibitory kind of response regulator. I would tell why it is inhibitory type of response regulator, because for final bioluminescence you must activate a molecule called Lux R. This Lux R is inhibited by this response regulator activity. So what this response regulator, which is Lux O, does, that it allows transcription of some genes, the RNA for QRR genes. These are the small RNA which are made, and they are complementary in a manner that Lux R cannot be synthesized or it cannot be transcribed. So when there is low levels of bacterium, you will have the signaling moving forward, a response regulator will be activated, and this response regulator will ensure that inhibitory RNAs are synthesized. which will ensure that lux R is not produced. So in normal condition, you will not have bioluminescence. And when I say normal condition, which means that the low density of bacteria. And this small RNA which is mentioned here, they seem to present a very delicate post-transcriptional regulation event which we see where RNA is removed by forming a double-stranded RNA-like situation by whether it is RNAi or microRNA and so on. So bacteria has utilized this strategy to ensure that the final activator is not activated or not synthesized. So how does bioluminescence happens in this case? Now when the growth of bacteria is happening and suddenly you have increased density of bacteria. This receptor, which was a hybrid kinase, is having an additional activity which is like phosphatase. So when there is a high density of bacteria, presence of these autoinducers as well as presence of additional autoinducers, they switch this receptor to in its phosphatase mode. What this phosphatase mode will do, that it will ensure that the subsequent lux U and lux O are not phosphorylated. And remember, the phosphorylated lux O was ensuring that these inhibitory RNAs are made. So when high density of bacteria is present, you will have phosphatase mode activated for the receptor, and that will ensure that phosphorylated lux U and lux O are not synthesized. What this translates into is that this inhibition is lost. High cell density. When this inhibition is lost, now lux R is synthesized and it can now go on and express the genes which are related to the quorum sensing. So basically what happened here is that right set of cues activated or switched the receptor from one state to the another state, from a kinase to a phosphatase state. Okay. So basically I can say that high cell density, which means that low lux O levels, particularly the phosphorylated one, which will ensure that high lux R levels, and this will ensure that you have the bioluminescence. You may like to go and see the work which is done by Dr. Bonnie Vassler on these species of Vibrii genus in terms of bioluminescence. So this will be the normal scenario, and this also remember high cell density, lux O level is low, and this is where you have the receptor as a phosphatase. And in case of low cell density, lux R was inhibited by QRR small RNAs. So QRR represents not just one small RNA but a group of small RNA which ensure that none of the molecules which are important for bioluminescence are synthesized. So let's see where else this two component system is used in microbes. If we think about bacteria, whether it is a E. coli, Pseudomonas, Mycobacterium tuberculosis, Staphylococcus or Klebsiella, all of them use this. And it is not just limited to them. These are some of the names which you may be very familiar with and can relate to, is that they use the two component system for multiple different activities. What those activities could be? The environmental conditions which is prevalent around them. Whether it is the presence of a metabolite like nitrate or nitrite, fumarate and citrate, these are some examples. There are multiple more. You can actually think about a metabolite, you can think about presence of an amino acid or presence of a compound, 4-carbon compound or sugar and so on, to which they need to respond, as well as the gaseous molecule like oxygen, carbon monoxide and nitrogen oxide, nitrous oxide. So what you notice is that whether the signal is of small type or some peptides or gaseous, whether it is solid, soluble and so on, all are actually ensuring that bacteria is responding to their presence. Sometimes metals, we know that some bacteria, they grow in very harsh conditions, like metals which are debilitating, having metals that have debilitating consequences on our health. Even those conditions bacteria grow. We also know that bacteria do grow in very high temperature conditions. So to respond to those conditions and remain healthy and maintain an equilibrium, the bacteria uses the two component signaling. And not to forget that some bacteria utilize this thing to metabolize some of the compounds which could be toxic or which can be utilized for other purpose. So whether it is methanol utilization, toluene, or sometimes the antibiotics which are given to bacteria, bacteria ensures that they respond to these antibiotics with the help of two component system. And this two component system in microbial world is not limited to only bacteria, but there are fungi, multiple of them. Some of them are like Aspergillus, Candida, they use this TCS type. Also remember that as the bacteria, you can always classify bacteria as very simple bacteria or very evolved bacteria or very complex bacteria, like for that matter Mycobacterium tuberculosis is apparently one of the difficult bacteria or complex bacteria. So more the complexity, you will see the change in the type of two component system, kind of switch from canonical to a hybrid type. We have seen example where both of them are functioning in parallel to ensure the response is mediated. Same thing will happen in fungi. You will probably see more of a complex or hybrid type of two component system working. They help fungi cope up with the stress responses or generate responses against the stresses like oxidative stress, osmotic stress or any type of nutritional stress. They also help ensure that fungi is sensing any change in the membrane fluidity or changes in the morphometric changes, in the sense that how fungi is looking or one form to another form. Then they will also help in the process of recognizing adhesion and moreover the virulence. Remember when we were discussing that if bacteria becomes sessile or when it forms the biofilm, it is no more as virulent as when it was free form. So even virulence, which is a property of these fungus, that is also affected, or rather the virulence properties are mediated by the use of two component system. Last but not the least, in case of fungi, the growth of hyphae, that is also being regulated by two component system. Conidia spore formation, we saw in case of bacteria, Bacillus subtilis, how spore formation was being regulated by two component system. Here also bacteria, in case of fungi, the Conidia spore formation is known to be regulated by two component system, as well as fungicide resistance, like we saw antibiotic resistance in case of bacteria. Not to forget that the cell wall composition of fungi is very different. So to regulate that and see if there is anything which is affecting the cell wall biogenesis as well as quorum sensing in fungi also, where two component system is utilized. So what we have understood in today's class is that the two component system in microbes is very important for responding to the environmental cues. It is widely used by bacteria in different processes. We saw quorum sensing today as well as sporulation. Earlier we saw how it is affecting the asymmetric cell division or life forms as well as chemotaxis. And you can see that fungi is utilizing this two component system in several stress responses as well as the spore formation which we saw in bacteria. Thank you.